I am, to introduce myself quickly, I am a carrot breeding senior with Rennes-Angers since a little more than a year. And before, I have been working more than 2 decades in crop research and genetics. And so I'm relatively new to the business and to the breeding of the carrot, and I still see it a little bit with the eyes from the outside, with the wider view, and I want to make today an appeal to the scientists in the audience and the students, which are, I've realized, numerous today, to please join carrot research, because it is thrilling, it is quite interesting. So my talk is called Nantes and Beyond, and I have co-authors. How does this look?
On the right.
Thank you. My co-authors are several breeders from Rennes-Angers and from our sister company Mikado Kiowa Seed, and also the pre-breeding. The carrot, raw or cooked, is part of the everyday diet in very many countries of the world. And when looking at the FAO figures, one can realize that the production has almost doubled since the 1970s, when the hybrids were introduced. The carrot consists mostly of water, but besides that, it is nutritious because it contains many important minerals and vitamins, and they are good for a healthy, balanced diet. This leads to what I call the carrot wheel, with the different players that participate in the today's situation. Let's begin at the top. No matter whether you want to turn it to the right or to the left direction, it's all possible. The scientists take their materials from the gene banks and study it and give the results onto the breeders and seed producers, which interact with the growers and the producers of the carrots and the traders in supermarkets today. And then there are, of course, the consumers, which we are all part of, and the conservationists. And all members of this rolling circle somehow interact with each other, depend on each other, and it's a giant wheel that is rolling. How has this all come about? It started with the landrace varieties that, since the appearance of the orange carrot, have been selected by local farmers. And just to mention these names: Flaky, Imperator, La Merveille, Chantenay, Nantes, Kuroda. These are all names that sound today, we are all familiar with them, well, most of us. And they come from the original landrace varieties, which I call morphotypes. What are the carrot morphotypes? The researcher Otto Banga published a book in 1963 where he catalogued all the major landrace varieties of the carrot, and he came up with 41 major such varieties, and he used only 2 classifying criteria: the vegetative period, the 3 levels, and the root size, with 5 levels. And altogether he had 7 morphotype classes identified, and when one looks them up, there are just 3 of these classes which contain virtually all nowadays used morphotypes. And these are the main crop with the long root, which are widely adapted, and the main crop half-long root varieties, and the early summer varieties. This is the start, and this is an illustration of it. One can see that there is a wide range of shapes of the roots of the carrot. The upper part comes from the seed catalog by Wilmereng and Andrieux and Company from 1870, I believe. So these traditional varieties have not changed very much, and nowadays they are maintained basically in the gene banks. So working with these landrace varieties, the modern varieties have been developed. And here we see an example for the Nantes hybrids of today, which look like the traditional Nantes from outside, but they can, of course, much more. Why? Because they have to be produced under industrial conditions. There are huge fields and there are huge machines, and these pose enormous stresses, disease stresses and also mechanical stresses to these poor roots. And as one can see, they are piled up into big packages. They are further packaged for the consumers after having been washed and brushed and weighed and sorted. And they must be matching the size of the bags where they end up specifically. They must provide for an optimal use of the volume that is available. They must have a nice appearance for the consumer, and they must, of course, be free of any blemishes and disease. So this implies a kind of challenge to the carrot breeders and scientists. Here's another example, the Kuroda varieties. They have a different shape, of course, than the Nantes varieties, and also slightly different internal characteristics. They are specific for the Asian markets. Early maturing main crop varieties, around 100 days to maturity. They are slightly darker orange from outside and inside. They also have a stronger, relatively stronger top foliage than the Nantes carrots. And although they are sown at a much smaller density per hectare, their yield is still considerable and has to be considerable. So this is important. They also have some weaknesses, of course, because they tend to bolt early, because they are not adapted to the cold seasons as the Middle European carrots are. They break easier because the roots are larger and softer, and sometimes there are problems with the shape. So these are all characteristics to work on. They also, of course, have to be apt to industrial production and marketing conditions. And my favorite of the tortures in the carrot world are the US-American imperator varieties of today. Look at these pictures. These oblong, slender roots are grown on large fields. They are harvested with heavy machinery, transported over long distances, then subjected to rather strong mechanical treatment to produce in the end these kind of baby carrots, cut and peeled pieces that you can see here. And such a carrot, I have read, travels on average 1,800 miles. This is equivalent to 3,000 kilometers from the field to the consumer. Now, what is all this about? Let's try to look inside. Let's try to look at the different traits which can be subsumed into complexes, trait complexes. And the preliminary survey yielded a total of 78 different traits which are spread out across such complexes as ability for industrial production, storability of the roots, adaptation to the cold, and there's a relationship with the vernalization requirement because the vernalization requirement of, the carrots are biennial, and in the second year they flower after having received some cold. This is typical for Western carrots. Some carrot varieties require a long cold period, others require a shorter period, and depending on this vernalizing period, they flower earlier or later. And this is, of course, a very interesting and important criterion for the marketing and growing of the carrots. We also have adaptation to day length and perhaps to heat stress, which could be related to what I dare to call summer bolting, which is different from the premature bolting after early vernalization. Then we have the complexes of disease resistance, either foliar or in the roots, or during storage or after storage, and down to the quality, including culinary ingredients and health-related substances, and the ability for, not to forget, the production of the seed, because this is an important part. Without seed, one cannot grow these crops. So 78 traits in total, and what I've seen so far and what was mentioned in this morning, there are several of these traits that have been investigated and some that have been under investigation until now, but the majority of these, and the list is available with me, probably have never been researched. And that's why this poses a big opportunity to researchers to go into the carrot research and do something for us. So now I want to give a second example that is related to the new the emerging new technologies. And here, of course, I refer to model crops and model technologies, which can and will eventually be applied in the carrot. This is my first of two examples on the phenotyping. I have been told that, particularly with the carrot, there are not so many qualitative traits. Most of the traits that one can consider tend to be quantitative. Now, what does this mean? Are the carrots really different from other plants? Are they different from other crops? And my answer is upfront, I apologize for my directness, is no. The carrot is a plant, as all the other plants also. The plants share the majority of their genes and pathways, and it is quite perceivable that the carrot also contains a lot of qualitative characters. Now, this example shows an experiment on 870 potato progeny, which were, of course, in replicated trials over several years, phenotyped for a disease, the late blight of the foliage, and this distribution here is somewhat, when one looks at it, somewhat binomial, but the curves are wide and not clearly distinct, and one could probably say that one part is more resistant and the other part is more susceptible, but they are mostly quantitatively reacting or acting. Now, a second criterion was added to the first. This is the amount of sporulation of the pathogen on the diseased plant parts, and the different colors depict this, and one can get already a step further by distinguishing more classes within these distributions. And finally, after repeating this exercise with two criteria, not by one isolate of the pathogen, but by 4 isolates, it was possible to indicate the specific resistance genotype to every single plant of these 870 plants. And it turned out that there are just 3 major genes segregating among this population, and they cause the different parts of this distribution, and the different colors here show that, in fact, when you look at the blue area in particular, these are the resistance, and the light blue, for example, refers to one specific gene. The phenotype of this resistance looks as if it were quantitative, because it covers a wide range of reactions, of responses of these individual plants, but in fact, it is genetically qualitative. And this is the difference which I wanted to mention here, and I believe this is very, very important for plant science and for the emerging technologies too. And here's one of them. If you have time, go to the Solarix webpage on CBIF, AT. This contains an index of virtually the total pool of resistance genes of the northern potato. And you have a browser. The browser allows you to visit every single resistance locus and the alleles that were found there. And the idea is that now the breeders can map the resistance they are interested in, find the R gene candidates that are related to this map position within the database, go back to the population, make markers, test the markers, and those that are useful, use immediately for breeding. So this will speed up the whole process. Now, to sum up this whole story, we have seen that the modern varieties of the carrot originate in the land races, which are mainly distinguished by their roots morphotype, and there are some specific characteristics that are related to each separate morphotype. Today's markets have specific preferences for the morphotypes. This has to be kept in mind. And more than 70 traits can be easily distinguished and must be researched and to be included better into the breeding. So there's rich opportunities for research. The carrot shares, of course, most genes in genotype environment interaction with all higher plants, and there are many qualitative traits and not only quantitative ones, although these qualitative traits show a wide plasticity in the phenotype, and the genomics will bring also now with the carrot genome at the verge new input to the breeders, and the methods how this can be used up will have to be developed by looking at the crop species. With that, I resume, and I'll take perhaps a question.